Hello everyone. Today we're going to be discussing cell reproduction. Now cell reproduction is a quite long process, so it might get a little tedious at times, but if you bear with me, by the end you'll be able to discuss the lovely life of a little cell and how you went from one cell to all the different cells in your body currently. So, to begin with, the overview of today's lesson in this video are the chromosomes and their role in cell division, to learn the cell cycle, which is basically the life of a cell, the stages of mitosis, the results of mitosis, how you control mitosis, and what could go wrong, and how to determine if there is a problem. And of course, as always, why is all of this important? So chromosomes are basically a single supercoiled DNA molecule with a protein, which is known as a histone. So when your DNA gets supercoiled, it becomes a chromosome. It becomes supercoiled because it helps it be protected. And when you're going to divide a cell, you want your DNA to be protected. And a chromatin, just for reference sake, is uncoiled DNA. So when DNA is actually used, it becomes uncoiled. It's kind of like you have a ball of yarn. To use it, you uncoil it. But when you want to keep your yarn together, you coil it up. Same idea with your DNA. And chromosomes are just supercoiled DNA. So you have DNA, you coil it, you get a chromosome. When it gets replicated, you end up with chromatids, which if you look at the picture, there's the X, that's actually two strands of DNA held together at the centromere. So you have two different strands, and that is a chromosome with two chromatids. One singular chromatid, if it's separated, actually is a chromosome, which gets a little confusing. But just think back to DNA is supercoiled, chromosomes are supercoiled DNA. So therefore it doesn't matter how much of it is in there, as long as it's supercoiled DNA, it's a chromosome. So you have strand of DNA, coiled chromosome. Once DNA gets replicated, you end up with two chromatids, which also equals one chromosome. Now, homologous chromosomes, on the other hand, are chromosomes that are not connected, but they have the same basic information. They have the same genes on them. And why you have homologous chromosomes is because you have a mom and a dad. So you got half of your chromosomes from your mom and half your chromosomes from your dad. And each set has a pair because you got one chromosome with eye color from your mom, one chromosome with eye color from your dad. So the chromosomes that have the eye color are known as homologous chromosomes. They're chromosomes that have the same genes on them, just one from your mom, one from your dad. And that is a major difference between diploid and haploid cells. Diploid cells have homologous chromosomes and haploid cells do not, which becomes more important when we discuss mitosis and meiosis, which you don't really know about quite yet. But when we get there, the diploid and haploid thing will come up. But in general, majority of your cells are diploid because you got mom and dad in there. Your haploid cells are your gametes or your sperm cells and your egg because you want sperm egg to combine and have the same number of chromosomes as your cells, so you need to have half the amount. So the cell cycle begins really anywhere, but if you begin it after cell division, you have a tiny little cell, and it goes into what's known as G1, where the cell grows. Then there's this G0 phase where the cell just does what it's supposed to do. It just lives. So that's where majority of the cell time is. And the R point is where it's kind of like the point of no return. If your cell goes past that R stage, then it's going to go fully through the cell cycle and go through mitosis, cell division. If it stays behind that R, then it can just stay and do its function. So the next phase is the S phase, where there's replication of DNA. So your DNA duplicates itself because it's getting ready for division, because you want DNA to be, all the DNA to be in each new cell, so you need the DNA to replicate itself. And then the next phase is your G2 phase, where the cell grows a little bit and prepares to divide. And then the M phase, which is mitosis or meiosis, and that's where your cell goes from one cell two more cells and divides itself. So cell division is actually a little different but depending on the cell, and it's basic enough to say a single cell becoming two cells. Prokaryotic cells go through binary fission, and basically because they're so basic and have nothing much to them, they have your cell, DNA replicates, and then you literally split in two and you end up with two cells. Eukaryotic cells, because we have a nucleus and all of that more complicated DNA, we go through mitosis or meiosis. An important phase in your whole cell cycle is your S phase, where your DNA replication occurs. And that's where you have your chromosome becoming a double chromosome or just another chromosome, but it creates those chromatids, sister chromatids. So it's when your DNA replicates. And they're, like I said before, attached by the centromere. So your two chromatids, your two strands of DNA, are attached to the centromere. And the important part of that is those two strands don't want to lose each other, because within this cell division, they're eventually going to be split apart. But if they lose each other, they don't really know what cell's getting what. So it's very, very important that those DNA strands hold onto each other.